Our first speaker tonight is Dr. Teresa Brentnell, who's an associate professor of medicine in the Division of Gastroenterology at the University of Washington School of Medicine. Terry has worked at the UW, has focused on the use of both basic science research as well as clinical medicine to improve the diagnosis and treatment of pancreatic cancer. Her team recently announced the discovery of the gene that causes familial pancreatic cancer. Dr. Brentnell and her colleagues are part of only a handful of people worldwide with the expertise in endoscopy, surgery, and pathology of the pancreas that is required for the early detection and treatment of pancreatic cancer. She's been a faculty member at our institution since UW School of Medicine since 1995, and she completed her undergraduate degree at Virginia Tech in Blacksburg, Virginia, and earned her medical degree from the University of Washington in 1987. She completed her residency in internal medicine at UCLA Medical Center in Los Angeles and served as a gastroenterology fellow at the University of Washington from 1991 to 1995. You guys, please welcome Dr. Brentnell.
I'm going to talk tonight. Now, did you guys go over the pancreas already in your coursework? That would be a big no. Okay, so that's a glaring hole there. And did you guys talk about familial syndromes that cause cancer? Did you go into that in depth? Yeah? Mm-hmm. Oh my God, I'm thinking another week is necessary for you guys to graduate. So I'm just gonna broaden your horizons. I'm gonna talk about pancreatic cancer. Why do we care about it? Does anybody here know anybody with pancreatic cancer? I'm gonna talk about familial syndromes. Why should you care about any of that? I mean, really, this seems like an obscure topic, but when I get done, you're going to be like, oh my God, how fun is that? Maybe not. Okay, now these findings are made possible by two sets of people. First of all, this research was done by 8 good-looking babes. These are my research collaborators. Here's me, look, nice hair day. And they come from all over the world, London and Pittsburgh, I mean, just you name it, they came from there. So it's really a collaborative work with the girls. And when we made our discoveries, we went out and had our nails done. So we feel like we did pretty good. The other part of research getting done is the fact that no research gets done without funding. Just doesn't happen, can't make a headway, can't find a cure, no treatment gets done without funding. And in particular, our funding for our discoveries on our work on pancreatic cancer was funded by people like you, people in the audience, people who care about the disease and gave us money to study it. We did not receive money from NIH or any public major care facility. This group, the Les Garden Foundation, helped us out, Canary Foundation, individual people just running a program that they chose to run themselves, and then a family helped us. All right. Now let's talk about pancreatic cancer for a minute. So first of all, how many people here know somebody with pancreatic cancer? Look at that. Oh my God. If you guys had eyes in the back of your head, you would see that most people raise their hands. So that's like a little scary. So as it turns out, 34,000 people die of pancreatic cancer every year. And 95% of those people die within the first year. So if you get the disease, pretty sure you're going to die within 12 months. Now that's terrible. It turns out it's the 4th most common cause of cancer death if you're over the age of 70. And it's the 3rd most common cause of cancer death if you're between the ages of 50 and 60. So it's common. It's a common cause of cancer death. Now the bad news is that if you came to me 10 years ago with pancreatic cancer and you came to me now, I'd pretty much be offering you the same drugs that don't work. So not much is new in pancreatic cancer, which is why it's so lethal. Moreover, it's a hard disease to work on, and let's talk about that for a second. So the pancreas is actually deep in your body. It's actually up against your spine, between your shoulder blades. So you cannot feel it on a physical exam. Cannot feel it. And the pancreas also is kind of a quiet little organ. It doesn't get gas, that's passing back and forth. It doesn't get diarrhea. It doesn't have a lot of symptoms. So by the time people get pancreatic cancer, it's pretty well advanced by the time that they get symptoms. So it's really advanced because it's an organ you can't feel, and it's an organ that tends not to get very many symptoms. Okay, so now what does the pancreas do? Well, the pancreas really has two jobs, two. One is to help you digest your food, so it makes little enzymes, and it squirts them out into your intestines so you can digest your food. And the other thing that it does is it makes the insulin, you guys have probably heard of insulin, makes the insulin that helps you control your blood sugar. So those are the two main things that it does. So those are important things, but they're not absolutely vital for you to live. You could take the pancreas out, you would become a diabetic. That's a person that doesn't have enough insulin, to control their blood sugar, so you become a diabetic. We could give you some of those enzymes. You eat them with your food so you digest it. So you can live without your pancreas. But in the meantime, it's just a little organ sitting in the back of your back, not making much trouble, paying attention to itself unless you get cancer. So we've kind of gone over the fact that most people have no symptoms, and the fact that by the time you discovered it, the disease has already spread rapidly to other organs. As if that was not enough, there are no good tests to find early cancer or precancer. So it's not like the prostate test, you get the PSA, your doctor might get that on you, see if you might have prostate cancer. We don't have a test like that for the pancreas. And then the final blow, as if that is not enough, is that it's a disease it's totally resistant to treatment, very resistant to chemotherapy. We don't have any good drugs for treating this disease. So what I'm going to do is I'm going to tell you a true story. It's a true story. This happened to me almost 10 years ago. It involves a clinic visit, a lost family, a killer, and persistence. So 10 years ago, I had a clinic visit. A 40-year-old guy came into my clinic, and he was concerned about developing pancreatic cancer because members of his family had the disease. And we dubbed this family Family X. And here's his family. So this is what you call a family tree or a pedigree. Everywhere you see a little black square is a person with pancreatic cancer. So here's my patient right here. So his two cousins have it, and his father, and 4 of his uncles, and his grandfather. So I was like, wow, you are in such trouble. But I didn't really say that, but I thought that, to maintain your decorum. So when I met this person, I thought, okay, I want to develop 3 goals. He really just sort of turned my vision of how I wanted my life to unfold in terms of what can I contribute? What can I contribute in medicine? So I came up with 3 goals based on this one interview with this one patient. Number 1, develop methods of early detection of pancreatic cancer. Number 2, find the gene that causes the disease in this family. And number 3, understand why is this disease so deadly? Why does everybody die from it? So 3 goals, 10 years. All right, so the first goal, in order to develop methods of early detection, we have to find the step right before cancer. And so it turns out there is a step before cancer, and we call that precancer, pre-cancer. And we grade it mild precancer, moderate, or severe. And severe precancer is like you're 1 step away from getting cancer, 1 step away. Okay, now tests to help find precancer. I'm a GI doctor, so these are GI tests. And these are 2 GI tests that we can do. One is called the endoscopic ultrasound. We have a little flexible tube with a TV camera on the end of it. We help you swallow it. It goes down into your stomach, which sits right on top of the pancreas. And then we turn on the ultrasound probe, and we can see the pancreas and see what it looks like and tell if it's abnormal or not. And then there's another test called a pancreatography, where we can put contrast into the pancreas and look and see if the pancreas looks normal and smooth like it normally should, or it's irregular and ratty and scary looking. Okay, so we can do those tests to tell us that the pancreas is not normal. But in order to make a diagnosis of precancer, we actually have to get a piece of tissue. We have to get a piece of the pancreas and look under the microscope. And so knowing that knowledge, we built our early detection plan. So first of all, in order to pass medical school, you have to be able to read pathology. So I'm going to tell you that this is a pancreas, And these cells are normal pancreatic ductal cells, normal. And see how these are called nuclei, the dark blue things, see how they're all the same shape and the same size, and they're all sitting with their little bottoms on this pink stuff that's called the basement membrane. So all the same size, this is normal. Now this is precancer over here, and these nuclei are all different shapes and sizes, and they're crawling all over each other, and their bottoms are not sitting on the basement membrane. And you are now pathologists because you can see that this is abnormal compared to this, which is normal. So that's precancer right there. All right, so our task at hand is to identify patients after they started down the precancerous pathway. We'll want to find them when they have precancer, but before the disease becomes invasive, before they get cancer. And the reason why is if they get cancer, as you know, they die from the disease because we don't have any good treatment. So we built a surveillance program, and we selected patients who we knew were at high risk for pancreatic cancer because their family has pancreatic cancer there, 2 or more family members with pancreatic cancer. And we started that program with this first guy who walked in the clinic. We've been running the program for 10 years now. We followed 75 patients from 50 different families. And so during that 10-year period, we found that 15 patients of the ones that we have followed had widespread precancer. And all of these patients were right on the verge of getting cancer and would probably be dead now. But we took their pancreas out because it was loaded with the precancer. So by taking the pancreas out, we prevent them from getting cancer, and they live to tell about it. Okay, so some of these people were from that family X. None of these patients who have undergone removal of their pancreas develop pancreatic cancer, so that's good. So it seems to be working. So goal number 1, check. Develop method of early detection. Now we're going to turn to goal number 2. How do you find a gene that causes disease? And why would you want to do that? Well, the reason you want to do that is if you can understand why a gene causes a disease, you can understand maybe why that disease forms. So I'm going to show you how you find a gene that causes a disease. First of all, you start with a large family. And in our case, we had that family X. And when you start with this large family, you're going to hunt for the gene. And so to know who carries the abnormal gene and who doesn't, you have to know who has the disease and who doesn't, okay? Who has the gene also gets the cancer. Those that don't have the gene don't get the cancer. Now, in pancreatic cancer, lots of people have found hereditary genes that cause cancer. Like people know about breast cancer is hereditary, and there's a breast cancer gene, so people all know about that. And there's genes that cause colon cancer in families, and there's colon cancer genes. But nobody had ever found a pancreatic cancer gene. And the reason why is because people who have pancreatic cancer are all dead, right? So you can't go get a blood sample because they're dead. So what helped us to be able to make this breakthrough was our ability to tell who had precancer. And by knowing who had precancer, we know, oh, you're affected and you're not dead, and we can get a blood sample. So this whole thing was made possible by the fact that the family X members were alive and available to donate their blood to help find the gene. So look, once this first guy came to me, the family began to trust doctors again, and all of these people that are in blue came in to get screened so I could figure out whether they had precancer or not. And the circles are the women's, and the squares are the men's. So over a period of nearly 10 years, these people all came in for their surveillance. And the people that are in red are the people out of this group that had precancer, and these are real people. And the people that are white did not have precancer. And we got blood samples from everybody, okay? So this is a large family, and now we know who has precancer and who doesn't. And we divided them up. We said all the people with precancer, we know you have the abnormal gene, you stand on one side of the room, and all the people who are normal and don't carry the gene, you stand on the other side of the room, and we're going to look at the blood between all these people and see what do they have that's not in common. What do they have that's not in common? Because what they have that's not in common is these people have a cancer-causing gene and these people don't. So they share a lot of things in common because they're family. But one thing they don't have in common is the gene that causes cancer. And we know these people all have it and these people don't. So we compare their DNA. So that's what we did. And we discovered there's a lot of chromosomes. People have 22 chromosomes, and then there's an X and a Y to help decide whether you're a girl or a boy. So we discovered that this gene that causes pancreatic cancer is on chromosome 4. And we kept looking and looking and looking, and finally, years later, we discovered that our gene is called pallidin. And I was as disappointed as you are to hear that it's called pallidin. I was thinking it would be called, something really cool like supersonic, super cool pancreatic killer gene. But no, it was just called pallidin. I was like, oh my God, that's such a disappointment. So it turned out that this gene has been described 5 years ago, but it wasn't described as causing cancer. It was described doing something else. But before I tell you about this gene, I just want you to know that not only was this gene mutated and abnormal in our giant family X, but we looked in people who had sporadic pancreatic cancer. That's people out in the community who get pancreatic cancer. And it turns out that this gene is behaving abnormally in those people too. So this is an example where when a gene causes a disease in a family, sometimes it turns out to give us information in the sporadic form of the disease that the general population gets. All right, so what we discovered is that this mutation in this gene, pallidin, tracks with every family member who got cancer or pre-cancer, and it was not present in any of the people who were cancer-free. And it turned out this mutation that we found was in a really important part of the gene. We also found that this gene behaves abnormally in sporadic pancreatic cancer, and this is an example of, here's our normal little duct right here, remember, with all the little nuclei sitting in a row, and here's precancer, and this is even more severe precancer, and this is cancer right here. And the more cancer-like you became, the more abnormally expressed this gene was. It's almost like it's a dose-related response. The more abnormal the gene was expressed, the more cancer-like the cells became. Okay, so we found the gene that causes the disease. So we've accomplished goal number 2. But so why do we care? What's pallidin? Why should anybody get excited about this gene? Well, it turns out it's kind of a cool gene. So remember those 8 good-looking babes I showed you earlier? Well, one of those babes actually discovered this gene 5 years ago, and she's a person who studies the cell. She's just interested in cells in general. And cells, in order to give them shape, have a skeleton just like we do. Like if we didn't have a skeleton, we'd just be a blob on the floor. And if you didn't have a skeleton, you can't walk. And that's the same thing true about a cell. So it turns out that pallidin is part of the skeleton of the cell. And when we talk about the cell, and we call it cytoskeleton. Cyto means cell. So cell, skeleton, cytoskeleton. So it's a protein that helps to make up the cytoskeleton. It gives the cell shape. It anchors the cell. Remember that normal cell I showed you where the nuclei are sitting on the bottom of the basement membrane? They're sitting down, behaving, and not crawling all over one another. So they anchor the cell down, and when appropriate, it helps the cell to move. Okay, so now if we put a mutation in this, like the mutation that we found in family X, what happens to the cytoskeleton? Well, this is what happens. So here is normal cytoskeleton in green. This is the normal gene, normal pallidin. And look what happens to the cell when you mutate it. So these family X members have cells that look like this. So their cytoskeleton's all gross and blobby looking. Not only is it gross and blobby looking, but you can tell when you look at these cells that these cells, which are hanging on to the bottom of the basement membrane and have a normal shape, are running amok over here. They don't have a normal shape. They're not sitting down like they're supposed to be. This is a kindergarten class over here. These people are out of control. All right, so the other thing that this cytoskeleton does is it helps the cell move. And cells moving is a key feature of cancer. So the way cancer spread and kills you, a cancer wouldn't kill you unless the cells got out and move around your body. I mean, really, any cancer, if the cells never move, you could just get that cut out and you're all done and you're fixed. It's the cells getting out and escaping. That's the part that kills you. So in order for them to escape, they have to move. They have to move out of the organ where they are and move into a different organ. And how do you do that? Well, that involves a cytoskeleton. Well, it turns out when we put this mutant pallidin into cells, the way it helps these cells move is it makes them grow feet. Now, how cool is that? And I don't mean like kind of like a vague kind of, weird idea of feet. I mean feet, like these, each of these are feet. Hundreds and hundreds and hundreds of feet grow in these cells. And now they can move. And they can move into places where they shouldn't be. This is actually the foot of a cell that's containing this pallidin mutation, and it actually is moving through a tunnel. It's actually moving through a tunnel, and we can actually, like racehorses, measure how fast these cells can move through the tunnels. And it turns out that cells with the pallidin mutation are 50% faster. This is a faster racehorse. So if you want to know how these cells get around, how they get around is the cytoskeleton. The skeleton, which is behaving abnormally, the cells don't sit down anymore, they don't maintain their shape, they grow feet, and they get up and walk. Now, how scary is that? Well, it's really scary. But the idea behind this is now that we know what's behind, what's driving the force behind pancreatic cancer, now we can do something about it. Let's make a drug that can combat this excess pallidin behavior. Let's make a drug that can prevent this pallidin from mutating. So to summarize what you've been able to learn tonight in your medical school career, you've been able to learn that mutated pallidin is a cause of familial pancreatic cancer. And normally that's a disease that's kind of rare and nobody would care about, except that it turns out that that pertains to the pancreatic cancer that the general population gets, the sporadic pancreatic cancer. We know that pallidin expression increases as the pancreatic cells become more cancer-like. So there's a direct relationship there, and if we could block that, maybe we could prevent the cells from becoming cancer-like. We also know that what the problem is that mutated pallidin is causing cytoskeleton abnormalities. The skeleton of the cell is misbehaving and becoming more mobile. Let's hope that this can give us a new idea, open the door for a blood test. So let's say that I could measure some things in your blood, maybe a pallidin level, and tell if you're going to be in trouble. Now, that's not something that's going to happen today or next week. But now we've got something that we can work with. We have some new ideas. Also, it's going to help us understand better, if we can understand how pallidin interacts with the other genes that are known to cause pancreatic cancer, we'll have a better understanding of how this disease forms. So it gives us a whole new area to start working on. It gives us potential new targets for therapy. So let's make a vaccine to prevent this from happening, or let's develop a drug that can block pallidin from misbehaving. And it gives us hope that pancreatic cancer is not going to be the lethal killer that it has been over the last 100 years and continues to be today. That's it. We take questions? No questions?
Stick around till after?
No.
No?
Okay.
No. You guys, because Dr. Brantnell's on call, maybe we'll take just 5 minutes to ask her some questions, and then we'll take a 5-minute break, and then we'll start up again. So come to the microphones, please, if you have a question.
Process you used to get from the sample from the blood to that slide that you showed?
To get to the gene discovery?
Right, right.
Yeah. Well, that's a really great question. So when people look for a gene, one of the things they do is, your DNA is long and vast, and contains, parts of DNA contain things that you don't really use. It's like your house. There's stuff in your house you don't really use. So having the DNA in humans has been sequenced by NIH. So the human genome, that's what we call all the DNA, we call it the genome. The genome is actually known, the sequence of the genome is known. And every few, let's say, if the genome was a string that went around the world, then let's say that every 10 feet there might be a little tiny piece of DNA that's not particularly important, but that varies from individual to individual. And so what we do is we're able to find that little piece of DNA, and we print it out for all the family members. Now, let's say that that piece of DNA makes up words. And we look at the words and we see, okay, here's the words for Susie. And we look on the page. And we look over here. Here's the words for George. And here's the words for Fred. And now we have a page on each patient. And then what we do is we take all the pages of the people that have the disease and put them over here. And we take all the pages of the patients that don't have the disease and we put them over here. And we see what are the words that these people share and these people don't have. And when we find the word, we look it up on our little genome database and find out where is the word located. And for us, the word was located on chromosome 4.
Yes, at the Mini-Med last year, one of the speakers talked about treating pancreatic cancer with HIFU. That was the acronym HIFU.
Yeah.
As of last year, the work with HIFU was just starting. Do you know how that's evolved and where that's going?
So HIFU is not a poem, you know, a 7-word poem. HIFU is high-frequency ultrasound. And this type of technology was developed in China. And originally it was sort of, it has a lot of promise. The idea is that we have this very high frequency ultrasound and that it can knock off cells in tissue, kill tissue. And originally it was sort of built around the idea, let's get rid of fibroids in women without having to do a surgery. But it became clear that there would be other applications, other ways that we could use this. So one of the ways that came up was, wouldn't it be cool if we could use it for pancreatic cancer? Because the pancreas being so deep in the body is very hard to operate on. And you know, it's a big operation. So that was one of the first applications people thought about. As of yet, the trials are not up and running, but they hopefully will be up and running probably maybe in the next 9 months.
I was wondering if your findings on how the cancer cells metastasize with that cytoskeletal abnormality and the extra feet, if you were able to generalize that to other kinds of cancer and how they metastasize too.
Yeah, that's a really good question. So the question is, you know, these cells grow feet. The cytoskeleton abnormality helps these cells grow feet, and then the cells walk away and start moving around and metastasizing. And so once we had our little breakthrough of discovering Paladin, we went back in the literature to see who else was working on Paladin. And it turned out that, this is a really cool thing, it turned out that they've been looking at it in breast cancer. And they did a really, 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 really cool experiment. They took a breast cancer tumor and they put it in a mouse. Now don't feel bad, the mouse wasn't that happy anyway. And so they put it in a mouse and then they put a pipette in with some growth hormone in it, and they said, we want to study the cells that can walk away, cancer cells that can walk away from the tumor. And as it turns out, most of the cells never walk away from the tumor. Only a handful of cells can walk away. And the ones that walk away have Paladin overexpression. So this is really cool because this is where one group of scientists discover one thing and another group's working on a different end of the elephant, and suddenly we begin to piece together how things happen. So in breast cancer, I think it does play a role, and we have to look and see what other cancers it might play a role in too.
Have you determined if people have to be homozygous for this errant gene in order to develop the disease?
Okay.
Or can they be a trait, just be carrying it and pass it on to their children?
Right. So that's a really great question. You've done very well in medical school, I can see.
No, I'm not a medical student.
So what she's asking is, does a person have to have, so you know how your mom and dad get married and then they have a baby? And this is how I explained it to my 17-year-old. Don't do that. So what happens is when that baby's being formed in the little uterus, in the womb with a view, is half of the DNA from the mom and half of the DNA is from the dad, and they get together, and that's what forms the new zygote baby embryo. Okay, so half DNA from the dad and half from mom. So at any time you look at a gene, any gene in your body, half of the DNA is from the dad and half is from the mom. So when we look at genes and the gene has a defect in it, sometimes you get a disease because both of these have to be abnormal. Like cystic fibrosis, both of these have to be abnormal, mom and dad. And sometimes only one of them has to be abnormal, and you get the disease. And this disease, only one of them has to be abnormal, and the other one can be normal. The other question she asked is, if this one is abnormal, do you always get the disease? And what's interesting is, in this particular family, I think you do get the disease if you carry the mutation. But I should let you know that probably multiple different genes may cause familial pancreatic cancer, just like familial breast cancer is caused by more than one gene. It can be BRCA2 or BRCA1. So this is probably caused by more than one gene. So not all familial pancreatic cancer is that lethal. And what I want to put across to you is, in a family that inherits a disease, just because somebody carries the gene mutation doesn't mean that they're always going to get the disease. Sometimes they don't. And it varies according to what the gene is. So you could have a gene where you're not likely to get the disease. So let's say BRCA2. Now, this is something you probably don't know, but BRCA2 in men, if you have a mutation in BRCA2 in men, you're more likely to get pancreatic cancer than you are to get breast cancer. This is the breast cancer gene. It actually causes pancreatic cancer. So what do we know about that? Well, what we know is lots of people carry the BRCA2 mutation. It's a relatively common mutation. And most people, most men don't get sick from it. Carry the mutation, don't get sick. But some of them do get pancreatic cancer, and occasionally people get the breast cancer. Now with this gene, pallidin gene mutation, most of the people who carry the gene do get sick.
So heterozygous then.
What?
Heterozygous, they can have a normal one, but that abnormal will take over. And I guess that makes sense if you just have one that's going.
One, so this one, it's this one, if this one misbehaves, it makes it so actually this one can't behave normally.
So you just have to look at the family history in order to warn parents that they might want to start. You just have to look at the family history in order to determine the risk of the
right.
siblings.
So you
and
look
the
at, so
children.
part of how you understand what your individual risk is if you carry a gene mutation is you look and see, is this gene, is it known to cause disease most of the time in most of the people, or is it known to not usually cause disease? So you look at what the gene is, and you look at the family history. So if everybody in the family has pancreatic cancer, like in family X, you carry the gene, it's pretty likely you're going to be in trouble.
Thank you.
Yeah.
One more question?
You mentioned the analytical process that you used to isolate pallidin as the culprit, and that it took 2 years to get through that process. Is that process fairly mature, or is it a developing science? And also, is it primarily human analysis, or is that something for which we're developing automated analysis tools and software?
So there were actually, I shortened this for you guys because I know that some of you brought your pillows with you and you're not afraid to use it. So, so the first part where we discovered it's on chromosome 4, that's standard fare. That's standard genotyping, gene discovery fare that people have been doing for, more than a decade, and they do it, on lots of diseases everywhere. So that's standard fare. Once we found it was on chromosome 4, it actually took us a long time to figure out which gene it was. We were able to, so going back to the page with words, we're actually defined a paragraph of words, and we didn't know which word it was. Actually, it was more than 250 words. We didn't know which word it was, and we spent a huge amount of time trying to figure out which one it was. And in order to find the word, we finally resorted to an idea I had about going back to the cancer tissue and using that to look for the gene, and that's how we actually found it. And it was just kind of a combination of a good night's sleep, common sense, and having a good day of coming up with that idea. Yeah. Okay. Thank you so much.